All right, guys. So, um, we're back for today. We're probably gonna be doing some biology. Uh, Biology, as I've said before, is pretty difficult for me. So we're gonna need to watch Yusuf Hassan video. I've already put the link to the video in the description. Shout out to him again for letting us watch it on the stream. Um, because I just really want to make sure that I'm showing the whole process. I'm I might not commentate as much, but yeah, we're just gonna be watching the video. Um I might laugh here and then, and make some jokes, but I don't know like how much commentary there really is gonna be. If you guys want to see the video, once again, link is in the description. Um and yeah, we'll get into the Anki afterward. Usually, as maybe some of you guys might know from last time, the the videos are split into two parts because chapter two, which is on reproduction, is 120 cards. It would normally already be split into 2 days. I really wanted to do organic chemistry today, but thing is that it's gonna be like it's gonna take too long to cover those 40. It's like 40 cards in in the first chapter on nomenclature. So I don't know if it's gonna be worth it to watch the video. Um, like I feel like it shouldn't. Hopefully, it's not that complicated. I'm probably just gonna try to like I don't know, just do it without the resource. Um, and yeah, maybe like next stream or the stream after that is when we'll try to do that. Um, and yeah, I think we can honestly after I do one last thing, we can we can get that booted up. It's already technically on the screen, but I just want to do one last thing. Um, all right. So, um, okay. Well, and make sure you guys can see that. All right, cool. All right. Let me also make sure you guys can hear it when I do
Okay,
this.
once again, hello everyone. We are live. Let's get started. Thank you guys for being here. Really appreciate everyone. We have a huge,
Make
very,
sure.
very full room. Kind of intimidating, can't lie. Um, Hope you guys are settled in. We have a pretty difficult lecture to give today. This is notoriously one of my least favorite chapters to teach. Number one, because there's a lot of content. Number
It's a
two,
really hard
I
chapter.
struggled with it when I was a student. And number three, it just kind of feels like me lecturing about something I don't truly know about. And that'll
That's
become
fair.
evident as we go on. This is chapter 2, general biology, reproduction. Every large thing is made up out of what? Small parts. Every large thing. Rocks, carpets, people, anything. Every large thing is made up out of small parts. And if you look back in time, there was a point in time where religion.
Oh, one second, guys. Uh, let me.
What are we? And from a reductive perspective, it seems easy to say that we're just human beings. But then you look inside human beings when they died, right? Or when they're alive and they got hurt, and you see there's things inside of us. We're not just like these empty cases of flesh that move around, right? So there are organs and tissue beds and muscles and this and that and blah, blah, whatever it is. And when we eat, the things just disappear, right? Where does it all go? What happens? So the question becomes like, you are a human, but you are also a brain and a heart and skin and eyes. And in the eyes are proteins, and in the proteins are atoms. And it's like, how far does this go, really? And in order for reproduction to occur, you can't just reproduce one thing and nothing else, right? Some things get produced, some things get reproduced, and some things get constantly destroyed and constantly recycled. And this happens throughout the body, right? Let me clue you into something. Your heart, the cells in your heart, the cardiac myocytes, right? You okay? Am I like, okay. Your cardiac myocytes, right? If they died, let's say you had a heart attack. By the way, what's a heart attack? Does anyone know? I don't know how a heart attack works. Yeah, but why? Your heart, let's take the box model of the heart. Your heart is split up into 4 chambers, and there are arteries that run along here in order to give your heart the circulation that wraps around behind, right? So there are sort of these arteries that come like this, and over time, because of diet and stress and smoking and this and that and blah, 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 you get a little bit of a blockage, and that blockage can turn to complete blockage, and then that will kill the oxygen supply downstream from that artery, which will choke out those cells, lead to inflammation, those cells will die, right? And eventually they will scar over. When you get cut, you scar over pretty well on your skin because your skin is capable of reproducing skin cells. I have a very large cut on my leg right here from when I was running track and field. A track spike went right through my leg during a race. But it scarred over pretty well. I have a keloid scar there, right? It's like a hypertrophic scar, right? But
I
it did
think you
scar over,
mentioned
and I
that
still
last
have mobility,
time.
and I can still like sort of feel the part around my leg and things like that. But when your heart scars over, it doesn't really regain function at any point. Why is that? It's because heart cells, and this is something that we didn't speak about before when we talked about cell types, these are permanent. They're permanent cells. They don't actually replicate, divide, grow in the classic way. The heart cells you have are the heart cells you get. That's it, right? Same thing with what to a point? 25 years old, right? What do I have that I have for the rest of my life? My brain. Neuronal plasticity is the ability for neurons to actually generate connections between one another. But do neurons duplicate and grow and regenerate? No. Well, I hope not, because there's such a limited, defined region of space inside the skull that if the neurons started dividing, you'd actually start to outgrow the space that you have inside your skull. That would lead to an increase in the intracranial pressure, and that comes with a bunch of different problems, right? But there are cells in the body, and the majority of cells in the body, I would be willing to say, that are constantly dividing. And this division that makes up the different tissue beds and organs and systems and blah, 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 whatever it is, this is what we know as cellular reproduction. And it is the
Imagine if you could enable
cellular
and
reproduction that
disable
we must talk about
neurogenesis. That'd be
in order to
crazy.
look at the big picture, right? Because almost everything inside of anatomy, physiology, this, that, the other thing, is taught from a perspective where it's like, well, here's the big picture and let's look small. The reason for that is because they assume that you come in with the knowledge of what does the small look like, right? And that's why we started with the cell in the first place, and that's why the chapter before this was the cell, right? By the way, for the people watching online, this chapter gets very nitty-gritty into endocrinology at the end when we talk about reproduction from the perspective of the menstrual cycle, which is going to be a completely different part of the video because I like to post that by itself because it's so heavy in content. I would heavily suggest that you watch my Fundamentals of Endocrinology lecture, which I will link in the description. And if you can't find it in the description, please just go looking for it. It should be right there. It is imperative that you watch that in order to fully understand everything that's going on in this lecture, as well as chapter 5 of biology, which is the endocrine system. Okay. In order to talk about cellular reproduction, we need to talk about the cell cycle. Cell cycle and mitosis. Correct? This is a cell from my cheek, a cheek cell. Right? I scrape the inside of my cheek with a wand
Buccal.
and I put it under a microscope, right? If I were to get a strong enough microscope to sort of look at the inside of this cell, and let's just say that it was in the process of replicating, right? Or at any point, it's a cheek cell, right? We don't know what sort of cell we call this, what subtype. This is a kind of weird question, but what type of cell do we call this? We have 2 different types of cells in the body, which have 2 different numbers of chromosomes within them. The somatic cell,
Uh,
right? A
oh,
somatic
I thought I was gonna say like
autosomal cell, correct? You'll find that there's a lot of like sort of
I was gonna say
double names
diploid.
inside of medicine and biology and things like that for whatever reason, bless you. And these cells are diploid. Meaning, diploid, listen very closely, of the necessary genetic material to code for the genome, it has 2. Of the necessary genetic material to code for the genome, it has 2. We are not talking about the stranding. We are not talking about the way that it does that. We're not talking about what genes are turned on, turned off. Just saying that for the necessary completion of the genome, it has 2 of those copies. Correct. So, in humans, how many chromosomes are needed to code for the genome? 23. 23 chromosomes are needed to code the entire human genome. Am I talking about single double-stranded? No, I am not. 23 chromosomes. And if you get 1 copy of 23 from your mom and 1 copy of 23 from your dad, that would make 46. This is what we call the 2n number. The 2n number is 46. Correct? Yes? So when cells exist within the body, and they're not dividing or they're not turning into germ cells, which are sexual cells, which pass on and recombine and create offspring, right? They have how many chromosomes in them? Louder. No. How many? 46. They have 2 copies of each of the 23. I just told you, somatic cells are diploid. And if we need 23 in order to code for the genome, and diploid means you have double the amount needed to code for the genome, that means that you have 46. Take a fly, for example. Let's say that there's a fly out there who has 2 chromosomes in its entire genome. That fly floats around with 4 chromosomes inside of it. One a pair 1, one a pair 2. I like to do this sort of demonstration. It takes a while, but let's say that you have chromosome 1 and chromosome 2. That's not a good way to show it. Chromosome 1, chromosome 2, and chromosome 3, and chromosome 4, and chromosome 5, blah, 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 all the way down to 23, correct? You have a pair of each one. I have a pair of chromosome 1, and a pair of chromosome 2, and a pair of chromosome 3. One came from my mom, one came from my dad. Does that make sense? We need to lock this in order to understand what's going to happen later, correct? I'm going to leave that there. Eukaryotic cells replicate through the cell cycle, correct? And this is basically a series of phases that the cell goes through where DNA is replicated, split, and the cell divides. You hear me? Yes? Yes? I need validation, guys. I didn't get enough as a child. Come on.
Yeah.
Am I in frame? There are much better diagrams of this in your textbook, in the Kaplan textbook. What smart person can tell me what M stands for here? Exactly. There are 2 things you need to remember.
Mitosis.
The cell is either doing one of 2 things. Either dividing or it's not dividing. Mitosis is the act of cell division. So everything that is not mitosis is not mitosis. Very simple. We have a name for not
Wow.
mitosis. What is it? Interphase. Very good. Interphase. So we have interphase and we have mitosis. And judging by the diagram that tells us that G1, S, and G2 are collectively known as loud. Come on, guys, there's a lot of you in here. Good, very good, fantastic. Interphase is the longest part of the cell cycle. It goes through these motions, and interphase, when a cell is not preparing for, when a cell is preparing for division, aside from when it's not doing that, which we'll talk about later, is the longest part of the cell cycle. It prepares for mitosis, and basically that's all it does. By the way, for anyone who's new here, you guys can get up, leave, eat, sleep. This is a very, very free environment. I don't care if you're eating in front of me. Just enjoy the lecture. That's it. That's all I'm here for. I'm here for your comfort and accessibility. All right. Cool. Interphase, like I said, is the longest part of the cell cycle, correct? Cells that are not dividing will exit this cycle and go into, what is G0? It is basically just where the cell hangs out, right? People sometimes, when they're older, they're like, especially doctors do this a lot because they have the liberty to do so. They sort of just like pick up and pack up whenever they want, hopefully, if you have that sort of schedule. They're like, yeah, well, I just quit my last job. But, oh, what are you doing now? Like, nothing. Just chilling. Living my life, right? G0 is the cell chilling and living its life, right? Does all the cellular processes, does everything it has to do, does its job to maintain itself, right? But it's just not working towards dividing right now. When you're in G1, S, and G2, you're kind of working towards dividing, correct? And the reason for that distinction is not random. It's because of how important mitosis is, right? Because of how important mitosis is. The entire time leading up to my educational journey to medical school is leading up to what's happening this upcoming fall, applying to residency. Every single one of the past 21 years, whether I knew it or not, was leading up to this fall. That's what it was all leading up to, right?
So G0 is like a
So
gap
it's
year,
almost like that.
realistically. I get it.
So we can say that the combination of G1, S, and G2 is interphase. And within interphase, individual chromosomes, for example, let's look at the fly again, with 2 chromosomes in its entire genome, correct? Which means that it has a pair of 2 chromosomes each. Sorry, it has 2 pairs of chromosomes, which makes it 4, right? These individual chromosomes are what?
I was about to be really confused if you
Not
didn't say that.
visible. Because they exist in a less condensed state. They're not actually going to look like that. They're going to be this weird tangled mess of DNA that you're not really going to see inside the cell, correct?
DNA spaghetti.
And the reason for that, who can sort of like deduce the reason that happens? Why isn't the DNA as tightly wound as it usually is? What part of the cell cycle are we in right now? Interphase. And during interphase, we're preparing for mitosis. And in order to prepare for mitosis, what do we have to do to the DNA? We have to replicate it. We have to make it over again. Imagine you're trying to transcribe a book, right? I made this distinction last time, and I think it's a distinction that doesn't go recognized by a lot of students. The difference between transcription and translation. Anyone here ever had like a teacher for another language, like at home, where they would make you like copy lines of like different, like stories and poems and things like that, and you just copy it? You look at it, you copy it, you look at it, you copy it, right? You've done that before to train your hand in writing that, right? Transcription is when you look at a language and you copy that same language. But translation is when you go from this language to one that other people understand. That's the difference between transcription and translation. So in DNA synthesis, transcription is actually done in order to transcribe the DNA and replicate it to get ready to divide later. Because the thing is, we want everyone to have the same amount of DNA, right? Let's say that I had a kid, and I had $100,000 saved up for this kid. And I was like, you know what? I'm going to make sure that each of my kids has $100,000. I have another kid. What do I have to do with my money? I got to double it, right? I got to double my money. So if you want every cell to have that same amount of DNA, you got to double your DNA, right? Okay, now, here's the question. If I said 2n equals 46, meaning that the diploid number is 46, we have 46 chromosomes in the body, when the DNA replicates, how many chromosomes are we going to have? Huh? 92 sounds like the right answer. Not really. It's still going to be 46. We're going to talk about why, right? Remember in the beginning I said I'm not talking about single or double-stranded. I'm talking about the number of chromosomes. Later on, when we talk about mitosis, we are going to talk about single and double-stranded. So if this trips you up, we will clear the air. Okay? All right. Cool. Everyone good on this? Any questions? Don't be shy. Promise. Yeah, less condensed. So let's say that I held up 100 strands of human hair like this from across a football field. Would you be able to see them? Probably not. If I took them and I crumpled them up into a ball and I held them on my hand, you might be able to. Maybe. Because they're going to take up a lot of dense space, correct? Something like that. So the more condensed the DNA is, the better you're actually going to see it through the microscope, correct? Okay, cool. By the way, brothers, it's Thursday night. We're going to dinner. Any questions about the chapter, about life, about your deepest, darkest fears and secrets? Anything? G1. Refer back to that diagram that I hope you drew or copied from the book, right? And if you guys need a professional diagram to mark up, like to put on your iPads, I want you to look up cell cycle Kaplan MCAT. That image will pop up. Cell cycle Kaplan MCAT, right? Is that for me? I'll take it in a second, yeah. I would really, really love to, but we're in the middle. So I will forego the pleasure of eating for all of you because you guys mean that much to me. Listen, if someone means more than food to me in my life, y'all are tripping. You don't understand. I'm a big back. Listen, when I say we're going out to dinner every Thursday, y'all better not flake on me, man. And if you do, I'm eating by myself. All right. This beginning of the chapter is a little bit boring because I know that like we're kind of split. Like half the people know this really, really well. There's like cellular biology majors and you've done this like 100 times. I promise, stick through, it gets better. And the fact that we're doing this now is actually going to make the end of the chapter a lot easier, right? The MCAT is a lot of patience and waiting. It's a lot of watchful waiting. You just look at yourself. You say, do I know it? Do I know it? Do I know it? And when the hammer comes down, you do, right? Okay. G1 is known as the pre-synthetic gap. The fact that it's known as pre-synthetic clues you into something about the diagram. G1 comes before S. So it's literally just the pre-S. And the G actually refers to growth. So this is pre-synthesis growth phase 1. G1, pre-synthetic gap, right? This is where all of the bulking is done. This is where you're putting on the pounds, right? The cell replicates mitochondria, which have what type of replication? What are the genetics of the mitochondria? Has its own DNA. Has its own DNA given to you by the mom. Every single mitochondria in every single cell in this room can be traced back to one female ancestor. A single one. The first female human ever. Correct? Okay. The mitochondria replicate. You also have the ribosomes, the endoplasmic reticula, etc. All these important organs, right? And because of all of this growth, you get an increase in size. This is the bulking, right? We have about an hour to multiply, right? Roughly? Okay. That should be good. And the passage from G1 into S is actually gated. You guys might remember my thing about the gated community. It's like, hey, listen, like, I know we live in the suburbs, but you're just not on my level, right? That's what the S phase is saying to G1. It's like, yo, I know you put in the work, you got rich, you got your money, you made sure that everything was done right, but this is a different tax bracket right here, right? There is a restriction point. any of you guys married? Anyone? You're married? Okay. When your husband does something, do you double check what he does? Sometimes? 100%, right? And it's not that you don't trust them. He's probably done it 100,000 times for her before, just out of his love for her. Inshallah, may Allah bless them. Say I mean. Right? He's probably done it a million times. He knows how to do it to the T. But it's not that you don't trust them. It's that you know what can happen if you don't check. You know what could go wrong. Right? Oh, patient's labs were fine. We just checked 4 times in the past 24 hours. Check again. 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 Because if you get it wrong here, you're done for. Right? I need you guys to really understand this point. It's critical. Absolutely critical that you understand the reason that a restriction point is there. A restriction point is there not because the cell is inefficient, but because it has so many things to do, and it executes them all so perfectly, and there's so much ahead that if one thing is out of place, then we're all done for. Right? There's a process in Islamic jurisprudence studies known as ijtihad. It reflects a serious and concentrated study and focus. There is extreme amounts of diligent prep work that goes into formulating a law. For example, a lot of modern Muslim jurists are asked, what is the ruling on investing in Bitcoin? Is it allowed? Is this something that we have to pay charity on? Is this something that we can put into our wills? Is this something people can inherit from us? Is it allowed in the first place, right? There's an immense amount of ijtihad that goes behind this, a serious study. This is the cell's ijtihad, the deep and serious study of what's happened. And you need to see if we can move forward from there, right? And the reason I'm making such a big point about this is because we're gonna see what happens when that thing goes wrong. When this fails. Horrible things. Okay. Are you all with me? Good. It's more of an answer that I like. This restriction point is basically to ensure that the cell has the proper amount, type, and accommodations of/for the DNA. You want to have a kid. You have food at home. You have money. Can you afford diapers? Do you have somewhere to keep a kid? Do you have good family support? Are you ready? That's the question, right? You want to replicate. You want to make a new cell. You have enough mitochondria, enough ribosomes, enough space, enough DNA. You have all the genes properly matched up. Is there anything wrong? Is this cell going to survive? And more importantly, is this cell that you're about to make going to kill us? That's the question. And who's us? Does the cell know that it's working as part of a whole body? No, not always. It doesn't always know that fact, right? Through hormonal regulation and through communication, it knows that there are other cells around and there are other things going on, but it doesn't always know the fact that it's part of something bigger, right? S-phase. Synthesis. So, remember how I said that the D phase, the DNA is open for transcription? Did I say replication? No, I did not. There's a difference between why the DNA is open in the G1 phase and why it's open in the S phase. In the G1 phase, it's open to code for the genes and the proteins that are going to make all of those materials that you're prepping. In the S phase, it's now open to be transcribed and replicated. This is the DNA synthesis phase, correct? This is where the cell replicates genetic material. Make sense? And this is so that, therefore, each daughter cell contains what? Identical DNA. You guys know how whenever there's someone who scores really well in the MCAT, everyone's just like, what was your study schedule? What did you do? How'd you do it? Let me do what you do. So when this cell that's lived its whole life and never had anything go wrong,
Hey, that's why I'm
survived,
here.
it's giving us
Hopefully.
exactly what it had. Because they said, it worked for me, it's gonna work for you. Identical. Same thing. Same circumstance, same DNA, same look, same habit, same environment, same everything. All the same. Correct? Why wouldn't it work? Correct? Right. I'm going to introduce you guys to a new set of abbreviations I use all the time. I use them everywhere. The A with the arrow and the P with the arrow. This means avant and this means post. Avant means before and post means after. So the P with the arrow means after, correct? Okay, so after replication, each chromosome, how many chromosomes? Human?
23.
46.
Oh, 20 pairs. Sorry.
Consists of 2 blank blank bound by a blank. 2 sister chromatids
It's like
bound
a
by a
centromere.
centromere.
Yes.
Centromere. You have a chromosome. Top and bottom. A chromosome. That in the G1 phase was open and replicating and doing whatever it had to do. When it enters the S phase, let's say the S phase occurs, it happens to it. This G1 chromosome that was single-stranded now looks like this. And it's not that those are crossing over the centromere. They're pinched together, right? So this is one side, and this is the other. Can you guys see that? You're taking this, you're taking the other, you're pinching them together. So this is one of them, and this is the other one. They're pinched at the center, correct? And they're held together like glue with these centromere. Does that make sense? Are you guys with me? Are you following? Because it's about to get very complicated. S phase happens. S phase happens. You guys all right? Good. So these are your sister chromatids. And I want to include these right here because I don't want you to get confused. Sister chromatids. Correct? Yes? Good. Copy that down. We'll take a second, we'll move on. This is very slow, very boring stuff. I know you guys probably aren't having fun right now. You will. In just a few short minutes. You'll be having a lot of fun, too much fun even. Yes? Sam, how are we doing? You good? Okay, perfect. Can I erase this? Yes. Yes. So G1, let's just say that G1 happened, the S phase happened, and then that got spat out. G1 happened, S phase happened, now that got spat out. And not even that you would actually see it in G1. It's not going to look like that in G1. I'm just showing you that it's single-stranded. Does anyone remember the name of the proteins that DNA wraps around in order to tighten up? Histones, which I previously mistakenly said contain histidine, but they actually contain the other two basic amino acids, lysine and arginine. Very good. You guys are smart. Smarter than I was. Can I erase this? So now we have sister chromatids. Now what? I'm going to tell you guys something that I'm going to say once, and I will take no questions about it. Because when people started asking questions, then I started to mess up. I'm going to say this one time. And for the people it clicks with, and for the people it doesn't, it doesn't. In the G1 phase, we have 46 chromosomes that are single-stranded. After the S phase, we have 46 chromosomes that are double-stranded. That means individual DNA chromatids, we have 92. Individual. And I got into a heated argument with a student because I once, and I stopped doing this, but I once called this 4N. And I stand by that. I stand by myself calling that 4N. Because if N is 23 single-stranded and 2N is 46 single-stranded, then 4N would be 46 double-stranded. You're doubling it. Talk about the number of specific DNA chromatids. 92 from 46 double-stranded. Does that make sense? Okay. I'm not going to answer questions about that. If it clicked for you, it clicked for you. If it didn't, it didn't. It's okay. It's not an important point. Welcome. How are you guys? Doing okay. From S to G2, we have another restriction point. Do you think that this restriction point is easier or harder to get through than the last one? Why? Why harder? Much more difficult to get through than the last one. Every single time you take an exam, right? You take the MCAT, take step 1, you take step 2, you take all the shelf exams, you do this, you do that. As you get closer and closer and closer, the exams become more critical. They're twisting the knife deeper into your head, being, no, answer. This is the question, answer. Now that you've replicated the DNA, anything could have happened there. Anything could have happened there. When you replicated the DNA, you could have swapped a C for an A, and now we're all screwed. Everyone's screwed. There is a scrutinizing, incredibly difficult position to get through right there. That is much more difficult than the last. As difficult as the last one was, this one's harder. Correct? The G2 stage occurs after the, and we call it SG2 checkpoint. Correct? Yes? How am I doing? Good. I mean that seriously, by the way. Occurs after the SG2 checkpoint. So the DNA has been duplicated. Have you guys ever noticed how I write the word the? It's like one big stroke. That, to me, that says the. My mom saw me do that one time. She's like reading something I wrote in philosophy. And I wrote the word the, and I said, yeah, the. You know, there's your T, your H, and your E. The. My friends describe my handwriting as a mix of script and garbage. And I say it's almost as though you took a spider, dipped its legs in ink, and let it run over the paper. I hope you guys tell me when I should rewrite or re-explain something. Okay? I am not a perfect individual. I might be very close, but I'm not perfect. Okay. And the cell here, the cell has a job. It's going to check the house. Like I said, when you're about to have a child, right? You're going to check the house. You're going to check the heating, check the walls, check to see if there's lead in the paint, check the room, check the money, check the bank account, check the food, check the this, check the that. Make sure that people are on standby in case you need them, right? Checking everything. Organelles, DNA, space, this, that, membranes, vesicles, everything. You're checking everything. You're doing your whole mental checklist. Anyone who ever moved houses before? Not like for college, but like actually moved homes. It's hard. It's really difficult. Really difficult. And as a human being, you're always going to forget something. Always, every single time. What did I forget back in New York when I came to Dallas? I forgot my running shoes. I had to buy a new $200 pair of running shoes. Annoying, right? But the cell doesn't forget. Definitely not purposeless. Not without purpose. Mhm. I want you to think that the cell is always capable of making mistakes. It just doesn't really happen all too frequently. And what I mean by all too frequently, I mean 1 in a billion trillion times. It'll happen. And then when it does make a mistake, it's caught by something else. Finally, after the G2 phase, you've checked the house, everything's ready, all the synthesis is done, restriction points have been passed, this is that. What do you think it's time for? Time to party. I have a habit, I don't know why, it's just like a sort of little ritual of mine. I never erase the title of the chapter, unless I need the whole board, right? In which case you will see me dramatically wipe off the title of the chapter. Very dramatic. I love the drama. I'm with all the drama. If you guys like being dramatic, do it. Mitosis. This is the M phase. You guys are always laughing about something. It's a very good question. Question was, if the cell is constantly replicating the DNA in the S phase and attaches them by the centromere, which is a protein or something the cell needs to make, does that also mean that while replicating, it is creating proteins and doing this and doing that? If the cell, let's take this for a ride. If the cell wasn't producing proteins at the time, what would happen?
I'm not gonna lie. I think we're just gonna start it. I think we're just gonna start downing people. I think we should just get started. Man. Well, before we do that, let's go get some water, and then we can continue. All right. 1 second. Let me fix my camera. All right. And we can do. All right. So. I feel like that's generally just like blasted off in terms of like how loud that is. Oh, it's because I have the volume loud, I think. Okay. Right. One second, guys. Sorry. All right. We're gonna change this to roughly like 60. I don't know why. Okay. All right. What is chromatin? Uh, Chromatin is that the condensed state of the chromosomes? Well, damn. Why did that say one day? One day. I never done this. Describe eukaryotic chromosomes and formation. This is weird. Why are the options one day? What? What? Hold on. There must be something wrong here, bro. Um, this is what I normally have. But why would this one? Why would this one? Oh, it's fixed now. Maybe. No, it's not fixed. Okay, so when I come over here, what is this one? What is this one set to? Have I just been using the wrong one this whole time, bro? basically made it so that if I press good, then it just moves on to the next. I mean, I guess. I don't know. Maybe that's genuinely, maybe it's genuinely for the best. I don't know. Um, I don't know. Large pieces of DNA formed by wrapping DNA around histones and packing the histones. How many chromosomes is the human genome composed of? 23. Oh, okay. I think I messed up that again. What does it mean to be diploid? It means that you have two pairs of identical chromosomes. I'm gonna get myself in again for that again. What does it mean to be haploid? It means you only have one copy of each chromosome. What are somatic cells? Um, they're cells of the body. What are gametes? Reproductive cells. Haploid sex cells. Okay. How is biological sex determined? Okay. How does the Y copy of the sex chromosome compare to the X copy? The Y copy is a lot like smaller. What is a centromere? Centromere is where I think it's the location. It's like the exact location where the two chromatids of one chromosome like are closely held. What is a kinetochore? That's the molecule I think that actually binds the two chromatids at the two sister chromatids at the centromere. Okay. Um Protein structures at the centromere serving as attachment points for microtubules of the spindle apparatus. assembly structures at the centromere serving as attachment points for microtubules of the. Okay. What are the four stages of the cell cycle? G1, S, G2, and mitosis. What is interphase? Interphase is any of the phases that are not mitosis. I don't even think I acknowledged it, but like earlier on in the stream, there might be a cut, and like just like ignore that part. Like nothing. Like I'm just gonna cut it out because realistically, there's not much. Like not like literally nothing happens. I think there's like a couple minutes of the Yusuf Hassan video, but it's like it's not worth leaving in. Um, I pretty much just called it a day at like the 1 hour mark. Literally like almost exactly at the 1 hour mark, and then we just switched into this. We didn't finish the video. The non-reproductive stage of the cycle. What is happening during G1? Cell increases size, synthesizes proteins and new organelles. What is happening in G0? Bruh, that was literally Yusuf Hassan's definition. Just living life and serving its function. What is happening in the S stage? It's replicating DNA. Okay. Does ploidy increase in the S stage? Um, I think it does in the S stage. Um, well, you now have four, so yeah. What? Oh, replication causes chromosomes to double in size. The two sister chromosomes still equal one chromosome, so the cell is 2N. Oh, damn. Yusuf Hassan led me astray. What happens in G2? Uh, I don't really know. I'm not gonna lie. More growth, more protein synthesis. Okay. Now this is when the cell starts like to split, like for real. What are the four stages of mitosis? Prophase, metaphase, anaphase, and telophase. Mitosis occurs or in somatic cells, obviously. What are spindle fibers? Is it just gonna go through all the new cards? Is that what we're doing right now? Are we just gonna go through all the new cards? Well, we're just gonna check this because I swear it is not bringing me to reviews for anything. Um, don't know. Let me check.
Several computers now have primary control of critical vehicle functions.
Okay. Yeah, I think it's just because of the intervals. So I think we might just have to we just have to set it up and see. It'll probably show up. The red card will show up eventually. Okay. What are spindle fibers? Spindle fibers are gonna be. That means that this whole time I haven't even really been using the proper like FSRS like algorithm. It's actually been using a completely different like I haven't even been applying to everything. What are spindle fibers? Spindle fibers are they're obviously like microtubule microtubule structures extending from the cent, central centrosome. Hold up, bro. We need to look up what the centrosome is again, bro. Yeah, okay. So I was right. It is. This is like the centrosome, but it has centrioles. Okay. What is chromatin? Yeah, it was just because of the intervals. What is chromatin? Chromatin is like it's it's just like the like unwounded form of DNA. What is an aster? Microtubules that anchor centrioles to the cell cortex. Um, okay, we're gonna change the word there because okay. Describe eukaryotic chromosomes and their formation. Um, they're like large pieces of DNA. Um, they're formed when DNA is like wrapped around histones and compacted. One one important things happen in prophase. Uh, what important things happen in prophase? Um, what important things happen in prophase? Uh, I don't know, bro. Chromatin condenses into chromosomes. The centrosomes migrate and form spindle fibers. The nucleus dissolves, and kinetochores appear at the centromere. Okay, guys, it's kind of bugging me that these steps are kind of annoying me. So just let me try to figure out how to fix this. Like I know how to fix it, but I don't know if I should fix it. Is my main problem. So I do have to change the settings. So, because right now it's at like it's just too high, bro. We cannot we cannot function off of those learning steps. I'm not gonna lie to you. It's just too high, bro. Okay. So, how many chromosomes? Well, 23 pairs. See, that makes a lot more sense. Those are the intervals that I like to see because I need those 1 minute intervals. I don't know if you guys know this, but how is biological sex determined? The X or Y chromosome. I'm just suspending this card, bro. I'll be honest with you. What are somatic cells? Any cell in the body that's not a reproductive cell. What does it mean to be diploid? It means you have two pairs of one chromosome. Not two pairs, two copies, bro. What is a centromere? Um, Centromere is it is the location where the sister chromatids are tightly bound. What are gametes? Gametes are reproductive cells. For males, it's sperm. For females, it's ova. How does that Y copy of the Y of the sex chromosome compare? The Y is like short, and it's mostly for like sex differentiation. So it relies on the X chromosome. If you're like a male, it would rely on mostly on the X chromosome for non-sex related genetic information. What is happening during G1? Uh, During G1, it's growing and synthesizing proteins and organelles. What is a kinetochore? It is the protein that tightly bounds the sister chromatids, or tightly binds the sister chromatids. Hold up. Does it not? Oh, I'm wrong. It's literally just the attachment point. Okay. What is happening in the S stage? What is newly created? Um, in the S stage, you have replication of the DNA. So you're creating a identical sister chromatid. What is interphase? Um, Interphase is uh, it's literally the period when, like, preparing for division, when it's generating like new organelles, replicating the DNA. What happens in G2? Um, protein synthesis. What happens in G0? Nothing. It lives its life and serves its cellular function. Does ploidy increase in the S stage? No. Although it's increasing the size of the chromosome itself, it is not increasing in ploidy since they're both bound together at the centromere, so it's still technically one chromosome. What does it mean to be haploid? It means that you have one copy of each chromosome. What is happening in the M phase? This is when you're actually dividing. All stages of mitosis, and it means you have two copies of each chromosome. What are the four stages of the cell cycle? G1, S, G2, and M. What are gametes? They're haploid sex cells. For males, it's sperm. Females, it's ova. Spindle fibers are microtubules that extend outward from the centrioles or centrosome. Um, what is a kinetochore? It is a protein structure that is found at the centromere, serving as the attachment point for microtubules to bind to and induce the separation of chromosomes during mitosis. What is interphase? Interphase is the non-reproductive part of the cell cycle. What happens in G2? More growth, more protein synthesis. What is happening in the M phase? Uh, it's essentially performing all the stages of mitosis and cytokinesis to produce a new cell. I guess you could say two new cells, but I don't think both of them are not new. One of them, one of them is still the same cell. It's the same, like at least mostly. It's gonna be at least mostly the same cell. It's like how many parts do you have to replace from a boat before it's a new boat? Like it's that type of thing, to be honest. What are spindle fibers? Um, Spindle fibers are they're microtubules that extend outward from the centriole in order to exert their function during mitosis. I don't know what was the definition I just gave, but I'm gonna give myself hard because I think I got that. Basically, these are microtubules that extend outward from the. They're like projections that go like all different directions from like extending outward from the centrosome in order to anchor that centrosome to the cell membrane. I don't really care that that makes the centrioles look like stars. How does the Y copy? The Y copy is irrelevant. It's little. It serves mainly for sex differentiation, and the X chromosome is mainly used for non-sex related genetic information. What is happening in the S stage? The S is the like DNA is being replicated. So you're creating an identical sister chromosome. What important thing happens in metaphase? Um, the formation of the metaphase plate. Yeah, that was pretty close. Spindles align, chromosomes align along the metaphase plate. Anaphase, that's when the chromosomes like separate. Like the chromatids in the chromosomes are gonna separate. Yeah, that's what I said. What happens in telophase? That's when cytokinesis begins and ends. So it like breaks the cell into two. Nuclear envelopes reform, chromosomes decondense, spindle disappears, and how you not gonna say cytokinesis, bro? That's like the most important part. Spindle disappears and centrioles return to their normal location. Chromosomes recondense. Okay. What is happening in G0? Uh, the cell is basically living its life. It's just performing its function. It's not working toward division. What important things happen in prophase? Um, uh, it's gonna the centrioles are gonna go toward like they're gonna start going toward the two axes of the cell, toward the opposite axis of the cell, opposite sides of an axis of the cell. I think is a better way of describing it. Um, then you're gonna have the nuclear envelope is gonna start disappearing. Uh, and what else? The nucleus dissolves and kinetochores appear at the centromere. Centrosomes migrate and form spindle fibers. Chromatin condenses into chromosomes. Okay. What are the four stages of mitosis? Prophase, metaphase, anaphase, and telophase. One important thing happens in metaphase. The formation of the metaphase plates. All the chromosomes align along the metaphase plate. So I should say the centromeres too. What is a centromere? A centromere is uh, it's like the part of the chromosome with two sister chromatids, the part where they're bound. What happens in telophase? Uh, in telophase, the nuclear envelope starts to reform. The chromatin starts to reform as well, or the chromosome starts to decondense, uh, and the centrioles begin to return to their normal location. How many chromosomes is the human genome composed of? Uh, 46. Yeah. Yeah, I'm gonna give myself a hard because actually, yeah. What is chromatin? It's the unwound form of DNA. What are somatic cells? Any cells that are not reproductive. What important things happen in prophase? Um, in prophase, the nuclear envelope disappears. The nucleus dissolves. The chromatin starts to condense. Uh, and the centrioles start to move toward like their axes. Oh damn, I forgot to form the spindle fibers. Yep. What is a kinetochore? It's a protein structure that you're gonna find at the centromere that forms it forms the attachment point of the spindle fibers that are going to attach during mitosis. One important thing happens in metaphase. Um, One important thing happens in metaphase. The spindles are gonna align the chromosomes at the metaphase plate. What is happening in the M phase? Um, Basically, all the phases of mitosis and cytokinesis to form two identical cells. What is happening during G1? During G1, there's protein growth and protein synthesis. Right. I meant cell growth. Oh, I forgot about organelles anyway. What happens in telophase? Basically, the reverse of prophase. So you're gonna have the nuclear envelope reappearing. You're gonna have the chromatin beginning to reform. You're gonna have the centrioles moving back to their original location, and the spindle fiber disappearing. Uh, and then you have chromatin, nuclear envelope. Uh, I don't know, bro. What else? Didn't I say that? I feel like I said all that. I'm gonna say I'm gonna give myself a good. I don't know. I think I got that. What are spindle fibers? Spindle fibers are the extensions from the centrioles um, that are going to I don't know. Like I know what they did. I just didn't. I forgot that it was that vague. Describe. They're large pieces of DNA that they wrap themselves around histones, and they can be compacted or packed. What does it mean to be haploid? It means you have two copies of each chromosome. Oh, bro. Oh my God. What happens? What important things happen in prophase? Um, in prophase, your nuclear envelope is going to be disappearing, and your your chromatin is going to be condensing. Then your centrioles are going to be moving to opposite sides of an axis, uh, and the spindle fiber is going to start forming. I don't think that. I think that's it. Oh, I didn't say that the kinetochores appear. Oops. One important thing is happening in anaphase. In anaphase, the cell starts to separate. Um, like they separate toward their like sides. The four stages are G1, S, G2, and M. Does ploidy increase? No. Two copies of each chromosome. What is is happening during G1? During G1, you're going to get cell growth and new proteins and organelles. They're haploid sex cells, sperm and ovum. What are spindle fibers? Spindle fibers are they're long microtubules that exert their function during mitosis. What does it mean to be haploid? It means you have one copy of each chromosome. What is interphase? What is interphase? Um, is the non-reproductive phases of the cell cycle. What happens in G2? In G2, you have more protein synthesis and more cell growth. It's just like it's small, and it's a small chromosome that's mainly serving the purpose of male sex differentiation. The X copy is going to be used for non-sex related genetic information. What important things happen in prophase? Your nuclear envelope is going to disappear or dissolve, and you're going to have the formation of the kinetochores. Then your chromatin is going to start to condense, and then the centrioles are going to start moving to their axis and start forming spindle fibers. What's happening in S stage and what is newly created? Um, what is happening in the S stage and what is newly created? So DNA is going to be replicated, and DNA is going to be replicated, and um, you're going to get an identical sister chromatid. What is happening in G0? The cell is living its life and performing its function. It's one of the microtubules that are going to basically extend from the centrosome in order to anchor it to the cell membrane. In the M phase, it's going through all the phases of mitosis and cytokinesis for the purpose of creating two new cells. What is the cleavage furrow? The cleavage furrow is like it's the place where the membranes start to pinch off uh, in order to separate between the two cells. It's kind of like a I don't know, like a cell butt. And it just starts breaking. Um, Not breaking, but cytokinesis is the splitting of the cytoplasm. What molecules are responsible for the timing of the cell cycle? Once I see this, I'm gonna remember it. Like cyclins and cyclin-dependent kinases. Okay. How do cyclins control the timing? It's the it's it works with something responsive factor. I think it's like P something. Um, it's like okay, so the it works with like P53, I think it's called, and those are at the restriction points. Damn, they didn't mention it. Rise or fall to activate or deactivate CDKs. CDKs. CDKs activate transcription factors that activate genes that allow cell cycle progression. What are the four stages of mitosis? Prophase, metaphase, anaphase, telophase. Um, it's the like pinched membrane that occurs due to microfilaments contracting the middle part of like what will become two cells. Um, what is cytokinesis? Once again, this is like the the microfilaments. The microfilaments are contracting in order to create like a pinching and eventually the formation of two cells. What molecules are responsible for the timing of the cell cycle? Cyclins and CDKs. 23 pairs. One important thing happens in anaphase. Uh, the um, the chromatids, like the spindles, pull chromatids, like separate the chromatids between the two like would-be cells. What is a kinetochore? It's a protein structure that forms at the centromere. Um, this is serving as the attachment point for microtubules coming from the spindle fiber during mitosis. How do cyclins control the timing? So cyclin levels are gonna rise or decline in order to regulate the timing. Those cyclins being there are gonna activate cyclin-dependent kinases that are gonna lead to the activation or deactivation of transcription factors that basically give the okay for the cell cycle to continue. What is chromatin? Chromatin is the unwound form of DNA. What important thing happens in metaphase? The chromosomes align across the metaphase plate by spindles. What important things happen in prophase? So the nuclear envelope is gonna disappear, and the chromosomes are gonna condense, or the chromatin is gonna condense. Um, then the kinetochores are also gonna form, and the centrioles are gonna start to move to like opposite sides of the cell. Oh damn, form spindle fibers. Bro, I always forget that. What are three quality control checkpoints in the cell cycle, and for what are they checking? You have, well, if there are three, there can only be three spots. There are only three spots. It's between G1 and S, between S and G2, and between G2 and M. They're basically checking for mutations, like to make sure that everything replicated correctly. Mm. I knew there weren't three in interphase, bro. Um, G1S checks if G1S checks if cell is prepared to synthesize new DNA. Does DNA need repairing? G2M is the DNA replicated correctly, and is it ready to divide? Metaphase and anaphase. is whether all the chromosomes have been attached to the spindle. So G1S is it ready to create new DNA? So it checks for repairs initially. It checks for errors in the DNA initially, right? And then once we get to G2M, now you need to check if it did it correctly. Then at metaphase, anaphase, it's like, okay, so everything was good, but like, is one gonna be left like in the middle? Like, if everything is not attached, then well, you could be screwed. What happens in telophase? In telophase is when you're basically everything reverse of prophase. So the nuclear envelope starts to reform, the um, chromatin starts to reform, the kinetochores start to disappear, as well as the spindle fibers, the centrioles start to go to their normal locations. Um, yeah, I think that's it. How do cyclins control the timing of the cell cycle? Uh, so cyclins are going to increase or decrease depending, like, in order to activate or deactivate CDKs, and CDKs are going to activate some of the transcription factors that activate the cell cycle or the continuation of the cell cycle. What important things happen in prophase? You're going to have the dissolution of the nuclear envelope. Then the chromatin is going to condense. The kinetochores are going to form, and the centrioles are going to start going to opposite sides of the cell, as well as the spindle fibers are going to start to form as well. I think that's it. Yeah. Large piece of DNA, they're going to form, they're going to wrap around the histones, and they're going to be packed with the histones. What is happening during G1? During G1, you're gonna have cell growth and synthesis of new proteins and new organelles. What are spindle fibers? Spindle fibers are gonna be It's been literally giving me only these cards for like ages. It really hasn't been trying to give me new cards because I had so many piled up from before. What are spindle fibers? Spindle fibers are uh, they're long microtubules that are going to extend from the centriole in order to exert their function during mitosis. So you have the G1S, which is basically the cell determining if the DNA is ready to replicate and if um, if the DNA needs any repairs, or if the DNA is ready to replicate. Then you're gonna have G2M, which is basically it checking if the DNA was replicated correctly. Then you're gonna have the metaphase anaphase checkpoint, which is gonna be if all the chromosomes were attached to spindles. What does it mean to be haploid? One copy of each chromosome. What is happening in the M phase? All of the phases of mitosis and cytokinesis to create to two daughter cells. When/what is the restriction point? The restriction point is it's kind of like a checking a point where you kind of you check to make sure everything is okay before moving on to the next step. Quality control checkpoint between G1 and G1 and S. If it passes through, it commits to replicating. If it doesn't, it goes into G0 or spends more time in G1 to try to pass the restriction point again. If DNA is found to be too damaged to move past the G1/S restriction point, what happens? So my guess would be that it would try to fix it. It would try to fix it or go into G0. It's like there's our P53. What medical condition can arise when the cell cycle becomes disrupted and damaged cells are allowed to reproduce? Cancer. The spread of cancer cells through the blood is referred to metastasis. Meiosis, gametocyte cells. What is cytokinesis? Cytokinesis is the it's like when the microfilament ring contracts in order to create the cleavage furrow and eventually the separation of the two cells. When/what is the restriction point? It's G1S. Um, in order to make sure that the DNA is ready to be replicated, if like it's halted by P53, if it goes in, if it passes the restriction point, it commits to the S phase and replicating DNA. If it doesn't, then it'll attempt to fix the DNA or go into G0. Like um, it'll spend longer in G1 to see if it can fix it and pass it, or it'll go into G0. What important thing? So the chromatids, sister chromatids, are separated between opposite ends. Um, well, then it doesn't. Number one, it doesn't get past the G1S restriction point. The P53 genes are going to basically stop that from happening. And then it's gonna try to repair the DNA to see if it can try to pass on the next time. What is a cleavage furrow? Cleavage furrow is kind of like the pinching of the membrane that occurs due to the microfilament ring that obviously pinches the membrane. Like, I don't even know what else to say. Are responsible. We have cyclins and CDKs. What is reductional division? It's a division that causes a reduction. That sounds. I have no further explanation. I'm gonna be honest with you. I don't know how it works. I just know that that's what happens. When the result of a division is a reduction. Why did I say it like that? Hold on. When the result of a division is the reduction of the ploidy number. Oh, like meiosis. Meiosis results in how many and what kind of cells. Um, so meiosis results in like from one thing is meiosis has too many phases, bro. Meiosis results in four haploid cells. was right. What are the main differences between prophase one of meiosis and prophase of mitosis? Um, Nothing. Homologous chromosomes undergo synapsis to form tetrads. They then undergo crossing over with one another. Um, what is reductional division? It's a division that results in a reduction of ploidy. What/when is synapsis? In prophase one of meiosis, it is the form like when they bind together to form tetrads, the two chromosomes with two chromatids each. So what where are the three quality control checkpoints in the cell cycle, and for what are they checking? The three quality control checkpoints are G1S is checking to make sure the DNA is ready to be replicated and it doesn't need any repairs. Then G2M is making sure that the cell is ready to replicate or ready to divide, and that the DNA was replicated correctly. Then metaphase anaphase is like, okay, are all the chromosomes bound to spindles? What are the main differences in prophase one? In prophase one of meiosis, the homologous chromosomes are going to form tetrads and cross over, while that doesn't happen in prophase of mitosis. How do cyclins control? So cyclins are going to increase or decrease in levels in order to activate or deactivate CDKs. And the CDKs are going to activate genes that are going to activate transcription factors that activate genes to determine whether or not to continue forward in the cell cycle. What important things happen in prophase? So the nuclear envelope is going to disappear. The chromatin is going to condense. The kinetochores are going to form. The kinetochores are going to form. Centrioles are going to start going to their like proper locations, and the spindle fibers are going to form. I think that's it. I hope I said the nuclear envelope thing. Cancer. What is a synaptonemal complex? Bumble. Yo, proteins that hold the homologous chromosomes together in the tetrad. Um, Proteins that hold homologous chromosomes together in the tetrad. Okay. What/when is a restriction point? It's at G1S. So basically, um, this is the point where it evaluates whether the DNA is ready to like replicate, whether it has any errors. If it does have errors, then P53 is going to halt it and um, allow it some time to basically make an attempt to try to repair the DNA so that like before continuing. What is a tetrad? It's when two homologous chromosomes are bound together. What is crossing over? It's like when parts of the chromosomes they just like swap locations. That's I feel like that's what I said. What is a synaptonemal complex? It's a protein that holds the tetrad together, like parts of the tetrad together. What is the chiasma? I have no clue. The point at which homologous chromosomes overlap one another. Oh, why can crossing over happen between sister chromatids? Uh, because they don't overlap. Oh, what happens in metaphase of meiosis? Uh, damn. Is it really gonna go through all of the steps of meiosis now, bro? Is that what we're gonna learn right now? What happens in metaphase one of meiosis? Uh, tetrads attach to the spindle apparatus at their kinetochores and align at the center of the cell. Tetrads attach to the spindle apparatus at their kinetochores. What is a chiasma? The place where the homologous, where the tetrads overlap, the two like separate chromosomes where they overlap. What/when is synapsis? Um, synapsis is it's when they form the homologous chromosomes form tetrads in prophase. Because they're identical. What is segregation in the context of a reproducing cell? Another term referring to separation. Okay. What is disjunction? Oh my god, bro. I hate this term. Separation of homologous chromosomes in meiosis. How is the way that chromosomes are split different in anaphase 1 of meiosis when compared to anaphase of mitosis? Um, daughter cells receive one chromatid from each chromosome. Daughter cells get daughter cells get one of the parent's two homologous chromosomes. What happens in metaphase 1 of meiosis? Uh, this is when the chromosomes, the homologous chromosomes, like the tetrads, they're gonna align in the middle. Oh my god, bro. What are the main differences prophase one of meiosis and prophase of mitosis? There's really no difference. The only differences are, in prophase one of meiosis, the like synapsis occurs, they form tetrads, and crossing over occurs. It means that the the chromosomes separate. Okay, I didn't say homologous chromosomes. What is this junction? The separation. It's the separation of the chromosome in meiosis. How is the way the chromosomes are split different in anaphase one? In anaphase of mitosis, you're gonna be 2N. In anaphase one of meiosis, you're gonna be 1N. In mitosis, you're getting one chromosome. You're getting one chromatid per chromosome. In meiosis, you're getting one whole chromosome. That's it. One chromosome with two sister chromatids. If DNA is found to be too damaged to move past the G1, well, P53 is gonna halt the cell cycle, and then it's not gonna be able to move past there. So it's either gonna go into G0, or it's going to attempt to fix the damage in order to try again. Results in four haploid daughter cells. What happens in my metaphase one of meiosis? The tetrads are going to align in the middle using the spindle fibers. What is ductional division? It means that it reduces diploidy. Like they divide, it reduces diploidy. What is segregation? The separation of homologous chromosomes in meiosis. Anaphase one of meiosis different from anaphase of mitosis. Um, it means that you have separation of homologous chromosomes as opposed to of chromatids within one chromosome. You're separating tetrads versus separating one singular chromosome. What happens in telophase one of meiosis, and how is it different from telophase of mitosis? Okay, it's the same. What is interkinesis? This is probably the the time between meiosis one and meiosis two. What happens in meiosis two? You're gonna get another cellular division that's going to break those two daughter cells into four daughter cells. Um, what medical condition can arise? Cancer. What happens in anaphase one of meiosis? They're basically the same, but anaphase one results in a reduction of ploidy. And telophase one is the same. What are the three quality control checkpoints? You have G1S, where it checks if the DNA is ready for replication and if it needs any repairs. Then you have G2M, where you check back on seeing if the cell as a whole is ready to divide, and if the DNA was replicated correctly. And then you have the metaphase slash anaphase checkpoint, which basically is going to you're checking if the chromosomes are all attached to spindles. What is interkinesis? It's the short rest period between meiosis 1 and meiosis 2. What is equational division? It means that there's no change in ploidy when it divides. I think the definition I gave was better. What slash when is the restriction point? The restriction point is at G1S. Uh, it's basically saying that um, it's checking back on the DNA and making sure that it's ready to be replicated and it doesn't need any repairs, regulated by P53. If the DNA is too badly damaged, then it's going to try to repair it, or it'll stay longer in G0. What happens in meiosis 2? Um, what happens in meiosis 2? Basically, it's going to produce four new daughter cells, uh, four haploid daughter cells. Um, and it's like pretty similar to mitosis. You're separating sister chromatids. What is the synaptonemal complex? It's the complex that was holding the tetrad together. What are the main differences between prophase 1 of meiosis and prophase? In prophase 1, in synapsis, they form a tetrad and they cross over. What is crossing over? It's an exchange of genetic information within the tetrad, like over the chiasma, across the chiasma. What is a tetrad? It's when two homologous chromosomes are bound together. Mendel's first law of genetics. I don't know. The law of segregation. Each organism has two alleles for each trait which segregate during meiosis. Thus, each gamete receives one allele. When the zygote forms, offspring will have two alleles, one from each parent. Which stage of meiosis is responsible for Mendel's first law? So Mendel's first law is the law of segregation, where the alleles are separated between, you're getting one allele from each parent. Which stage of meiosis is responsible? Metaphase 1. I mean, no, anaphase 1. Sorry. The alleles of a given locus separate into separate gametes. Why can't crossing over? Because they're identical, so you wouldn't realize. What is the second law of classical genetics? Inheritance of one trait has no effect on the inheritance of another. Crossing over in prophase 1 is responsible for this. Inheritance of one trait has no effect on the inheritance of another. Inheritance of gene for eye color has no effect on inheritance for hair color. Um, what is Mendel's first law of genetics? It says that the alleles are separated into their appropriate, like their corresponding gametes, like into different gametes. Each organism has two alleles for each trait, which segregate during meiosis. Synapsis occurs in prophase 1 of meiosis. What is a chiasma? The place that the two homologous chromosomes overlap in a tetrad. Which stage of meiosis is responsible for Mendel's first law of genetics? Um, Meiosis is responsible for Mendel's first law of genetics. Anaphase 1. What is disjunction? The separation of homologous chromosomes in meiosis. It says that the inheritance of one allele has no bearing on the inheritance of another. What stage of meiosis is responsible for his second law? I think it literally just said prophase 1. It's because of crossing over. Bruh, what? Um, um, I'm reading the Khan Academy to try to figure out what this law means. I just don't get it. Um, I always want to like explain to you guys what I learned, and and I feel like it's very often that I look it up and then I don't learn anything. And I just give up, and I'm just like, okay, I just accept it at face value with like the thought in my head, like, okay, like when I get to the questions, I'll like see how it's applied. Um, so basically what I learned is, so it's saying that in metaphase one, you're gonna have a like okay, so you're gonna have your chromosomes like this, right? And like this sound like okay, right? So if this is from mom and this is from dad, right? Sometimes like it can be the case that although this one was from mom and this one was from dad, that doesn't mean that the one at the bottom has to be the same order. Because it can also be the case that when these two chromosomes come to the middle, instead of being like this one is actually dad's and this one is actually mom's. So they can be randomly like aligned, which is going to like in the creation of your own gametes, it can be like, oh, mom's, dad's, like it doesn't matter. It doesn't matter. Like it's completely random which ones end up where. And that's already in addition to the crossing over that has occurred by this point already. What is segregation in the context? It's another term for the separation of homologous chromosomes. It's the law of segregation. It basically says that the alleles are you have two alleles, one from each parent, and they're separated. How is the way the chromosomes are split different in anaphase one, when compared to anaphase of mitosis? Anaphase one is going to result in a reduction in ploidy because you're splitting homologous chromosomes. What happens in metaphase one of meiosis? In metaphase one, you're gonna align homologous chromosomes and along the metaphase plate using the spindle fibers. In telophase one, it's the same. What is interkinesis? Interkinesis is gonna be the rest period between meiosis one and meiosis two. What is equational division? There's no reduction in ploidy. How is the anaphase one different from anaphase two? I mean, from anaphase of mitosis. Uh, Anaphase one is gonna be the separation of since you're separating. I swear we just did this. Since you're separating the homologous chromosomes, it results in a reduction of ploidy. What happens in meiosis two? In meiosis two, you're gonna create four. You're gonna create four haploid daughter cells, um, and that's pretty similar to mitosis. What stage of meiosis is responsible for metaphase one? since they can be aligned and they can be aligned differently. What is Mendel's first law? It basically says you have one allele from each parent, um, and these are segregated during meiosis such that each gamete receives one allele. What are the main differences of prophase one of meiosis and prophase? So in prophase one of meiosis, you're gonna have like synapses to form the tetrad, and you're gonna have crossing over. Um, no, not exactly, because of the existence of linked genes. What stage of meiosis is responsible for Mendel's first law of genetics? Um, Anaphase one. Uh, it's that the inheritance of one trait like is independent of the inheritance of the other. Um, Responsible for Mendel's second law of genetics. Metaphase one. No, because of linked genes. What is Mendel's first law of genetics? It says that like you have each organism has two alleles, but these segregate during meiosis such that each gamete gets one allele, and then the resulting zygote from reproduction is gonna have two alleles, one from each parent. All right, we did it. Okay, so 110, 2 hour 30. All right, so what we are going to do is I'm gonna pull up Jack Weston so that we can. All right, screen capture. Um, all right, all right. So, um, great, cool. Damn, this is another day. Another day. Every single day so far they've just been like, oh, sorry, you guys hearing that twice. It's okay. I need to pause it anyway. Um, I hope you guys weren't hearing that twice. Hopefully that wasn't annoying. All right. This is another day with 6 questions, bro. Oh my god. Okay, I was considering C, so the answer choice is correct incorrect because about this with the younger example. Yo, I was kind of concerned for this, bro. I'm gonna read all of them because I wasn't sure. Answer choice is incorrect because it goes beyond the passage information. The main idea of the passage is that what? Really? I wasn't sure, bro. It's true. You would be treated as differences as if there were similarities. But it's like there are all these different types of single parents, and you're classifying all these differences that they might have as under one main similarity that they're all single parents. I got this in like 2 seconds. I'm not. Yay, we got one wrong today. It's a good day. It's a good day indeed. They were all easy. Bruh. I took the longest today and they were all easy. The average is in 83. Most people got one specific question wrong. At least one question wrong. You're the one that I want. Ooh, ooh, ooh. Okay. So thank you to everyone for watching. Um, and I will see you tomorrow. Good night, good evening, good morning, good afternoon, and yeah.